Hey, 2050 students. Welcome to our second set of lecture material, which is all about cells and cell membranes and membrane transport. So I want to start out by establishing the fact that all life is cellular. Whether we're talking about human beings, whether we're talking about fish, whether we're talking about an amoeba, every single living thing is composed of cells. The cell is the functional structural unit of life, okay? And this is not a cell biology class, but we're going to get into some detail about cells and their components and their structures. And we're going to start out by talking about the cell membrane. So this is a depiction of a cell right here. There's its nucleus, some mitochondria, rough and smooth endoplasmic reticulum. And all these things, of course, are on the inside of the cell. And the cell interior is separated from the cell exterior by this structure right here, which is the cell membrane, also called the plasma membrane. And external to that plasma membrane, so outside the cell, we have extracellular space, which is going to be filled with extracellular fluid. And this space can be small, it can be big, the amount of fluid can be small, it can be large. Now, as far as extracellular fluid goes, there's two main types. There's blood, of course, which is in our blood vessels, and then there's interstitial fluid. An interstitial fluid is the fluid within tissues, so it's also called tissue fluid. It's between cells. So here I have a bunch of cells, you got like 10 cells up here, and they're not packed super tightly together, so there's some space between them. This would be extracellular space, and this extracellular space will be filled with extracellular fluid, in particular filled with tissue fluid or interstitial fluid. Now, blood, on the other hand, would be found inside this blood vessel. So interstitial fluid is going to surround and bathe cells, tissues, and organs. Now, what's inside the interstitial fluid? Well, we'll have a lot of electrolytes, sodium, potassium, calcium chloride. We'll have nutrients like glucose, wastes like ammonia, gases like oxygen, CO2, and of course it's a fluid, so everything is dissolved in water. Now, one of the jobs of blood is the flow of blood is going to basically replenish and regulate the interstitial fluid. And by making sure our interstitial fluid is okay, is in homeostasis, well, we can make sure the interior of our cell, our intracellular fluid, is also okay and in homeostasis. All right, good stuff. Now, inside that cell, we're going to have organelles like our mitochondria, rough and smooth endoplasmic reticulum. We've got our nucleus too. And everything inside the cell is referred to as the cytoplasm. We got organelles, we got intracellular fluid. Now, the plasma membrane, we know it separates the inside of the cell from the outside. So it is separating extracellular fluid from intracellular fluid, two separate fluids, okay? If we look at the plasma membrane, the cell membrane, it's going to have two main components, lipids and proteins. We know a little bit about lipids and proteins from the last lecture. Now, the lipids in particular, they're the most abundant lipids in the cell membrane, are these lipids called phospholipids. And this is a depiction of the structure of a phospholipid here. It's kind of like a balloon with two tails. And the phospholipid has a phosphate group, that's the round head here, and it's got two fatty acid tails. And one of the important consequences of this structure is the head is hydrophilic. So the head is hydrophilic, it loves water. Meanwhile, the tails, they're hydrophobic. They hate water. And this actually is really important for a functioning membrane, to have a part that is both hydrophilic and a part that is hydrophobic. So because of this nature of the phospholipid, when we make a membrane out of phospholipids, they have to be arranged in a bilayer. Remember, the inside of the cell has intracellular fluid, which is mostly water. The outside of the cell has extracellular fluid, which is also water. And because the phospholipids have a hydrophilic part and a hydrophobic part, we got to make sure hydrophilic part is facing inward and the hydrophilic part is facing outward. So this is why we need a double layer, a bilayer. That way we can have hydrophilic heads facing outward, hydrophilic heads facing inward, and the tails are in between. So this is actually really good. It lets us separate the water on the interior from the water on the exterior. So our cell membrane is a phospholipid bilayer. Now, not only is it a phospholipid bilayer, but it has proteins either embedded within it or associated with it. It's got some other stuff that we're going to see as well. Now, this collection of phospholipids, proteins, and other things, including cholesterol, that represents our cell membrane is actually called the fluid mosaic model. It's fluid because everything is not exactly locked in place, as we're going to see. It's a mosaic because it's made of many small different parts. All right, let's go into a little more detail on some of these components. We mentioned the phospholipids already. Notice the phospholipids are in a double layer, a bilayer. The purple guys here are the phosphate groups. They're hydrophilic. Notice how the hydrophilic part is facing inward, facing the intracellular fluid. And we also have the other layer with the hydrophilic part facing externally, where we have the interstitial fluid. Then our fatty acid tails are on the interior of the membrane here. Okay, so I mentioned again that the plasma membrane is fluid. And it's fluid because the phospholipids are capable of some movement. Like a phospholipid can spin around. This phospholipid could diffuse over here. Really, the only thing phospholipids can't do is they can't switch. Like this guy is on, part of this layer facing the exterior. He cannot then go and become part of this layer facing the interior. That's the only thing phospholipids cannot do. Now, this fluidity is important because think about a cell that's going to divide. If a cell divides, so here's a cell. If a cell divides, the membrane is going to go inward. The membrane goes inward, and then the membrane pinches, and we got two resulting daughter cells. But the membrane can't go inward unless it's fluid. Also, if I go back for a second, let me erase all this stuff first, then it'll let me go back. If I go back for a second, if we look at these proteins in here, we actually can put proteins in and take proteins out. And when you put a protein into the membrane, the phospholipids just kind of get pushed to the side a little bit. When you take a protein out, the phospholipids are going to then move into that space where the protein was. So this fluidity is really important. It's important for cell division. It's important for anytime a cell has to move the membrane, like if it's doing phagocytosis. It's important for the ability to add new proteins in, take new proteins out of that membrane as well. Now, the plasma membrane is a mosaic because it's made into many different parts, and we see many different labeled things here. We've got phospholipids, we've got a variety of proteins, we even have some cholesterol in here, we have some things called glycolipids and glycoproteins too. So lots of different parts, that's what makes it a mosaic. Now, cholesterol actually stiffens and strengthens the plasma membrane. It prevents it from becoming too fluid. If it's too fluid, it wouldn't have that structural integrity that it needs. But what about those proteins? There's two main membrane protein types. There are integral proteins and peripheral proteins. Now, this guy right here is literally going through the membrane. So what word do you think is more appropriate if it's literally going through the membrane? This one also literally going through the membrane. Well, these two guys, they are both integral proteins. So integral proteins are embedded in the membrane. Now this guy, he's not embedded in the membrane. He's definitely associated with it. And so he's known as a peripheral protein. He's a peripheral protein right here. So two main types, integral and peripheral. And integral proteins, one way we can describe these guys, these two here, is they are transmembrane, meaning that they go across the entire membrane. And as we're going to see, this is going to let these proteins have some really neat and important functions. Speaking of protein functions, there are 5 basic kinds of integral proteins, and all these types are related to their jobs. So there are proteins that are transport proteins, receptor proteins, cell adhesion proteins, structural proteins, and proteins that are acting as enzymes. Let's go over these 5 different types. Transport proteins allow material to enter or exit the cell. Some transport proteins are channels, like this guy right here. This guy is a channel protein. He has a passageway down the middle. These molecules are able to move from the outside of the cell to the inside of the cell. Notice how they're moving from where there are more of them to where there are fewer of them. So they're moving down what's called a concentration gradient. Now transport is really benefited by these channel type proteins. As we're going to see, channel proteins are really crucial for hydrophilic things to get through the membrane. All right, but channel proteins are not the only way proteins can do transport. As we're going to see, there's a couple other ways too. We'll get to that in a moment. But for now, let's look at some receptor proteins. This is a receptor protein. So the receptor protein is binding to an external signal. An external signal is called a ligand, and the ligand is binding to this receptor protein. And it's not depicted, but the receptor protein is going to cause something to happen. There's going to be some sort of response. Maybe this protein here will break off and do something, maybe a piece of it, maybe this part will be activated and it'll do something, but there's going to be some sort of internal cellular response. Receptor proteins allow an external signal to create an internal response. Okay, what about some other jobs? They can be cell adhesion proteins. So here I have 2 neighboring cells. So I got cell number 1 and cell number 2. We know that cells come together to form tissues. Well, sometimes we're going to need to physically attach the neighboring cells together. And so I got one transmembrane protein and it's being grabbed onto by another one. And these are called cell adhesion proteins because they're adhering cells to one another. And like I said, this is crucial for the formation of tissues. Okay, there's also structural proteins. Structural proteins allow cells to create and maintain their unique shape. Shape is very important. Think about a cell like a red blood cell. Whoops. A red blood cell has this shape. It's called a biconcave disc. So this red blood cell has a shape, a biconcave disc, and this shape is maintained often by structural proteins. Okay? Sperm cell has a characteristic shape as well, and that shape is maintained by structural proteins. And of course, structure and function are totally correlated with one another. So structural proteins are really important because if a cell loses its shape, it can lose its function. All right, we've also got enzymes. Enzymes catalyze chemical reactions, help turn one chemical into another, help break things down. Not every enzyme is a membrane protein, but many of them are. All right, cool. So that's it for our different functional types of proteins. Let's talk about another way we can describe proteins. There are structures called glycoproteins. A glycoprotein is a protein, so we got this membrane protein here, this blue guy, and he has a sugar group attached to him. So this is a glycoprotein. Notice I also have a glycolipid over here. So this is a phospholipid with a sugar attached to him. And these glycoproteins and glycolipids, they play a big role in cellular recognition. Especially if your immune system has got to know what cells are yours and what cells are not yours. So like your combination of glycoproteins and glycolipids are going to be something that identifies your cells as being yours. And so a cell with a different combination would be considered foreign and your immune system would kill it. Now we actually have a word called the glycocalyx. This refers to the entirety, the collection of all those surface glycoproteins and glycolipids. This is a close-up of some microvilli on a plasma membrane here. So here's all these different microvilli. We saw these in the epithelial tissue lab. And then this fuzziness here is the glycocalyx, which is all the sugar proteins and sugar lipids on the cell. And like we said, it plays a role in immune recognition, but also in terms of being a protective surface and in adhering cells to one another. Okay, so we're done with our structure of the cell membrane for the most part. We're going to want to now talk about how things can go through the cell membrane. And what a cell membrane does is it exhibits something called selective permeability, meaning only certain things can directly pass through it. So oxygen and CO2 can zip right through the cell membrane. Non-polar molecules, and by the way, non-polar is just another way of saying hydrophobic. So non-polar molecules, hydrophobic molecules, like steroids, for example, can go directly through the cell membrane. But water and polar molecules, and polar is just, as you might have guessed, polar is another word for hydrophilic. So hydrophilic molecules like glucose cannot go through. Ions like sodium, potassium, calcium, etc. cannot go through. So only certain things can go directly through the membrane. However, as I'm sure you've realized, well, we might need to get ions and get sugar and get water to go through our membrane to get these things into or out of the cell as needed. But these things can't go directly through the phospholipid barrier. So how do we get certain things in? Well, we're going to use membrane transport. And there's actually two big types of membrane transport. There's active transport, and that's where the cell spends ATP. ATP is our cellular energy, of course, and the cell is going to spend ATP in active transport. Then there's passive transport, where the cell is not spending ATP. So these are my two basic types of membrane transport. Like we just said, passive transport does not require the cell to spend energy to spend that ATP. So in this case, because we're not spending energy, substances are simply diffusing. So they're going from an area of high concentration to an area of low concentration. Things that are in solution are going to diffuse from where there's more of them to where there's less of them. And diffusion is going to be a type of passive transport. Active transport requires the cell to spend energy. And here we see an ATP molecule that's going to be broken down, and the energy from that is going to be used to move this blue guy from an area of low concentration to an area of high concentration. So active transport, you're going against that gradient from low to high. In diffusion, passive transport, you're going down that gradient. Now, a good analogy is passive transport is like going downhill. If you're on a bike and you're about to go down a hill, that's the best, right? You just cruise down the hill, you coast down the hill, you don't have to pedal at all. All right? However, going uphill is a totally different story, right? You have to exert energy in order to go uphill. So if you're going from high to low, that's like going downhill. That's going to be passive. If you're going from low to high, that's like going uphill. That's going to be active. Now, again, to reiterate, passive processes driven by diffusion, where things are moving from a high concentration, those little brackets mean concentration, by the way, to a low concentration. If I drop some dye in a beaker of water, right here, the concentration of dye is going to be very high, and of course it's very low, it's nothing around here. And so the molecules, because they're always moving, they start to spread out. We still have an area of high concentration here, and we've got some of the dyes spread out, but still low concentration here. And diffusion keeps happening until there is no more concentration difference. The dye is still moving, but now every time one moves to the left, another one moves to the right, so everything's balanced out. Now, if I just see another example of diffusion here, high concentration on this side of my cell membrane, low concentration here. These molecules are able to go directly through the membrane, so they just start doing that. And they'll diffuse through the membrane until the two concentrations have equilibrated. These blue guys will still be moving, but every time the blue guy goes this way, another one goes that way. Okay? Now, the speed of diffusion depends on the size of the concentration gradient. If you have a bigger and bigger concentration gradient, the diffusion happens faster and faster and faster. All right, so just remember this proportionality here. Diffusion also depends on the temperature at which it's occurring. And if we try to get something to dissolve and diffuse in water, if the water is hot, the diffusion, the spreading out will go a lot faster than if the water is cold. So diffusion depends on molecular movement. Molecular movement is going to depend on kinetic energy, which is going to be influenced by temperature. Okay. Another thing that influences diffusion is the size of the molecule diffusing. Small molecules diffuse very quickly. Large molecules diffuse slowly. So the bigger the molecule it is, the slower it's going to diffuse. Okay, good. Another thing that influences diffusion is how thick the thing how thick the membrane is that it's diffusing through. So in our lungs, this is a tissue of lung here. You might remember this epithelium from lab. And in the lungs, oxygen is diffusing through a few cell membranes. CO2 is diffusing through a few cell membranes. And these are some of the skinniest cell membranes that we've got in the body. And that's good because it enhances that exchange, that diffusion. It's going to take longer to go through a thick membrane than it will a thin one. And that just makes sense. All right, in this example here, I've got some transport going on. I got some purple guys going directly through my cell membrane. I got some orange guys going through a protein, some blue guys going through a protein, and some more orange guys, orange diamonds here, also going through a protein. Which of these four situations is depicting active transport? Well, it's got to be the one where we're involving ATP. And in this example, we're going from an area of low concentration to an area of high concentrations. That's going uphill. That requires energy in the form of ATP. In all three of these, we're going from high on the outside here to low on the inside. So in all three of these, we're doing diffusion. Can you figure out which of these three guys is the non-polar one? Well, remember, only non-polar molecules can go directly through the membrane. So the non-polar one in this case is the purple one. All right, good stuff. We're going to talk about diffusion a little more, including something called facilitated diffusion. So there are two types of diffusion that occur through the cell membrane. Two types of diffusion that occur through the cell membrane. One type is called simple diffusion. The other type is called facilitated diffusion. So let's go through those a bit. In simple diffusion, the substance is just going directly through the membrane. In simple diffusion, the substance goes directly through the membrane. In facilitated diffusion, a transport protein is required. So here is a transmembrane protein. It's a channel, and the green guys are using it to go through the membrane. So in facilitated diffusion, there has to be a protein facilitating, helping the process occur. So facilitated diffusion will involve a transmembrane protein. Simple diffusion, no protein is necessary. The molecule is simply going right through the membrane. So substances that undergo simple diffusion include oxygen. We see oxygen diffusing from where it's in high concentration outside the cell to low concentration inside the cell. We see CO2 diffusing from high concentration inside to low concentration outside. So CO2 and oxygen can go directly through that cell membrane, and that's simple diffusion. Hydrophobic things, in other words, non-polar things like steroids, as well as your fat-soluble vitamins, vitamins A, K, E, and D, they are going to simply diffuse as well. They can go right through that cell membrane. No transport protein required. Okay, facilitated diffusion can occur via channels. So here we have a channel protein. This channel protein is called a sodium leak channel because it allows sodium to diffuse into, in other words, to leak into the cell. Notice sodium is going from high concentration on the outside to low concentration on the inside. This charge on the sodium, it's got a positive charge here. It's a cation, so it cannot go through the membrane. It doesn't like that hydrophobic interior there. So sodium has to go through a channel. Same deal for potassium. Here's a potassium leak channel. Allows potassium to diffuse from high concentration inside the cell to low concentration outside the cell. And these channels, these openings, there's going to be water in here, just like there's water outside and inside. And the ions are happily going through a watery channel, whereas the ions cannot go through this hydrophobic interior here. Okay, but channels aren't the only way we can do facilitated diffusion. We can also do carrier proteins. Carrier proteins are kind of like channels, but they're a little bit different. In a carrier protein, the protein grabs onto the molecule on one side, so here's the glucose, grabs onto it on one side, then it changes shape and releases it on the other side. So in a channel, there's no shape change. It's just a pore through the cell membrane. But for a carrier protein, there is going to be a shape change. And a great example here is this glucose carrier protein. Now, one of the things that we got to be aware of is that channels can be selective. So a channel can have a particular size to it. So if you look at your periodic table, you look at where sodium, potassium, and calcium are, they're of different sizes. So we can restrict ions based on how big they are by adjusting the pore size here. We can also adjust the charge in here. Maybe we can only allow positive ions through, maybe only allow negative ions through, maybe only allow, monovalent ions with a single charge, or versus divalent ions with two charges to them. But we can be selective. And selectivity is important, of course, for homeostasis. Now bear in mind, carriers can be selective as well. Like this carrier protein, look at how it has this like circular shape within it to bind to the molecule. So it's not going to bind to a molecule of a different shape, a different structure. So carriers can be quite selective as well. And what's interesting is carriers can end up being saturated. So if I only had one carrier protein, well, I can only move one molecule through it at a time. There's only there's a maximum speed I could do this facilitated diffusion with a carrier, because I'm only going to have a finite number of these guys. If I only have one, and it takes a second to move this guy in, I can only move one per second. Doesn't matter how many I've got out here. If I've only got one carrier protein, and it takes a second to move something, I can only move one per second. All right, so carrier proteins can become saturated or overwhelmed. Like, it wouldn't do me any good if I just kept adding these yellow circles out here. My carrier protein couldn't go any faster. Now, we can actually represent this in a graph. We said earlier that concentration gradient, as it gets bigger, the rate of diffusion gets bigger as well. And this is true up until a point for facilitated diffusion through a carrier protein. In facilitated diffusion through a carrier protein, we're going to reach a point where the carrier proteins are all saturated. They're all working as hard as they can. They're all occupied. So it doesn't matter if we add, more and more solute. We're going as fast as we can. Okay, good stuff. I'm going to quit right here. We're going to separate this cell membrane lecture up into a couple parts. We're like 33 minutes in. I think that's a good spot to stop. When we meet next time, we're going to pick up, talk about osmosis. Then we're going to talk about active transport and a couple other things. And then we'll be done with this topic. All right. If you have questions, of course, seek me out. And I will see you later. Bye-bye.